Hello everyone and welcome to our LIPID MAPS webinar today. It will be presented live by our guest speaker Hubert Schaller, entitled Sterol, Metabolism and Homeostasis in Plants. So we're very much looking forward to that. My name's Ruth Andrew and I'm based in the University of Edinburgh and I'm going to host today. We expect the webinar to be around about 45 minutes and we'll allow 10 minutes for questions at the end. So I'd like to introduce our speaker to start with. Hubert defended his PhD at the Université Louis Pasteur in 1992 on the selection and characterisation of tobacco mutants resistant to inhibitors of sterol biosynthesis. This was under the supervision of Professor Benveniste, and he did a postdoc at the Plant Molecular Biology and Genetics Department in the Max Planck Institute in Cologne in Germany and in 93-94. Before that, he worked as a visiting research associate in the University of Malaya in Kuala Lumpur in 89-90 in the field of botany and phytochemistry. And that was on a programme on a new phytochemical survey of Malaysia. So he was then hired as an CNRS scientist in 1994, where he's developed his research on plant isoprenoid biology there since 2006 as a team leader. So the goal of his team is to provide a refined understanding of critical roles of steroid lipids and the regulation of sterol pathways in plant biological processes.
Yeah, thank you very much, Ruth, for the invitation to talk in this Lipid Maps webinar series, and thank you for the very kind introduction. What I would like to do now is to share a little bit of the research we have been doing the last years and also more recently about sterol biosynthesis in plants. So I'm a plant biologist, trained as a biochemist, and I'm working in Strasbourg in the east of France, close to the German border, in a research institute called Institut de Biologie Moléculaire des Plantes, so Plant Molecular Biology Institute in Strasbourg here. And the first question I would like to discuss with you is a little bit like why it is so interesting to work on sterols in non-model organisms. And the reason is that, all these sterols that you can find in non-model organisms, in non-mammalian organisms, are very different structurally speaking. So they are different from cholesterol, which is the main mammalian sterol, but is also a sterol quite widespread in plants and other organisms like red algae, for instance, Chondrus crispus, with 90% of cholesterol in its content, in its profile. And they're also very different from ergosterol, the major sterol found in fungi. So this diversity is mainly linked to the structure of the side chain. I have a number of examples here I have put with red structures on the side chain. So you can have 1, 2, or 3 additional exocyclic carbon atoms, exocyclic regarding the cyclization of the C30 precursor. We will see that in a moment. So you can have 24 methyl, 24 ethylidene, 24 ethyl sterols, some algae like dinoflagellates. They have dinosterol with this particular structure here with a methyl group at position 23, an extra methyl group at position 4, and then you have structures with unsaturations, position 22 or position 24. And then you have an additional level of different structures. It is linked to the stereochemistry at position C24, where you have epimeric pairs of sterols like cleonasterol and cytosterol. So all these compounds are generated by pathway with interesting enzyme, and in some of these non-model organisms, there's a kind of golden mine to dive into it. So this is an astonishing diversity of the sterolome of photosynthetic organisms and beyond. So in the next slide, get a little bit more focus on another layer of diversity. So that's the diversity of sterol pathways. So some organisms make their sterols from acetyl-CoA through the mevalonate pathway, through which the C5 building blocks isoprenic units are formed in these organisms. And some other organisms they make their isoprenic units with a completely different chemistry with respect to acetyl-CoA. So they use glyceraldehyde phosphate and pyruvate to make methyl erythritol phosphate, which is the so-called non-mevalonate pathway. And so from those precursors, you can have different branches and different biosynthetic segments, starting from cycloartenol or lanosterol to end up with the pathway end sterols, like 24-alkyl sterols in embryophytes, in seed plants, which I will talk about now in the next part of the presentation. The main questions and objectives are twofold. Like, one is about the cellular function of phytosterols, so the multiplicity of the sterols found in plants, using Arabidopsis as a model, and what are the key regulators implied in isoprenoid phytosterol homeostasis. So that's the two main questions we are working on. And so the next slide is discussing the sterol C-methyltransferases. So this is a class of enzymes that participate to the diversity of the sterol profiles you can find in plants and other organisms. So basically, there's a family of enzymes with distinct functions. So they're adding exocyclic carbon atoms at position C24, and one is sterol methyltransferase 1, which is using preferably cycloartenol as a substrate to generate 24-methylene cycloartenol. And then further down in the pathway, you have another methylation reaction that is occurring at C24-1, according to the sterol nomenclature. And so this reaction is catalyzed by enzymes called sterol methyltransferase 2. And so in Arabidopsis, there is a genetic redundancy, and there are two enzymes responsible for this reaction. And this is defining a branching point at this stage. So that's 24-methylene lophenol, the substrate of the SMT2 classes of enzymes, producing 24-ethylidene lophenol as a product. And then you have a branching point here, which goes to 24-methyl sterol, like campesterol, and 24-ethyl sterol, like cytosterol, and then stigmasterol with the desaturation at position 22 here on the structure. So campesterol has been discovered in 1996 as a precursor of brasinosteroids. Brasinosteroids were known since long, but the biological function that came out through Arabidopsis genetics is only quite recent. I would say this was known through seminal work done by Joan Corey and some others, showing the biosynthesis of brasinosteroids from campesterol. So we did some work on the functional analysis of sterol methyltransferases. Yes, we were able to show that genetic inhibition, so using knockout mutants in both sterol methyltransferase 2 and 3 genes in Arabidopsis, is impairing growth quite severely. So on the picture here, you see on the left a wild type individual of Arabidopsis thaliana. So that's the favorite model plant for those who don't know the plant world so much. A species closely related to rapeseed. And on the right side, you have one of these double mutant individual that is poorly growing, but forms a little rosette. So the rosette is a structure, the botanical structure of this species. But growth doesn't reach further developmental stages like bolting, flowering, seed setting, etc. So you can see on the right side of the slide, you have the wild type total ion current GCMS analysis. You have classical sterol profile of Arabidopsis wild type plants with a number of compounds, the two major one being campesterol. In fact, campesterol has these two epimers, which are not separated, of course, in GCMS, and then you have cytosterol. And you can see, I hope you can see on the completely right, you have a peak of the mutant, so you have a huge peak of campesterol, and it's almost no cytosterol. I cannot say there is no cytosterol, but it's close to to zero. There's a little tiny peak of cytosterol. So I mean, these individuals here are completely devoted of cytosterol, and this is asking the question about the function of cytosterol. Yes, so we did this work in collaboration with an outstanding collaboration with a colleague of mine since quite a long time, Daisaku Ota at Osaka Prefecture University, now called Osaka Metropolitan University. And we were going a little bit further into the characterization of this growth impairment in this Arabidopsis mutant, and were able to show that the cell division is impaired. So this is these photographs are showing embryos of Arabidopsis dissected from seeds. And you have the division plane, which is going into wrong orientation in the mutant compared to the wild type. So for us, that's the key information about the possible role of cytosterol in growth and development, starting from cell division, and that's the working hypothesis for what we are currently doing, trying to figure out how this is working and what is the dedicated role cytosterol may have in plant development beyond its role as a membrane structural component, of course. So the other part of the things I wanted to talk with you, so it's about the key regulators or regulators implied in isoprenoid and phytosterol homeostasis. There are some interesting information we gathered first a few years ago, studying cycloartenol synthase, the first committed enzyme in the sterol pathway, so using 2,3-epoxysqualene as a substrate. And so Cas1, cycloartenol synthase 1, also coined oxidosqualene cycloartenol cyclase, is producing this typical triterpene, steroidal triterpene, bearing a 9-beta-19 cyclopropyl ring on the cycle B of the sterol. And then cycloartenol is converted into pathway end products, as I presented earlier. So we were able to analyze a series of knockout and knockdown mutants that we get from several libraries of insertional mutants. And one interesting mutant, which is described, yes, is a knockdown, is a weak allele. It's a partial loss of function cycloartenol synthase 1,1 allele that has a morphological phenotype, which is quite surprising and still surprising, although it's a number of years that we have isolated this mutant. So you have on the left side of the slide, you have a wild type Arabidopsis. So it's a close-up on stems bearing calyx, the fruits. And then next to that photo, you have a photo of the mutant. So the mutant is basically having the same phenotype, except some layers of the stem. So epidermis layer and chloranthema layers, which are shown on the section. You have a section with white and with green and white cell layers. So the chloranthema in this mutant has lost its photosynthetic capacity, and you have plastids, chloroplasts that are drastically modified in the mutant, with most probably very high, very big plastoglobuli present in this damaged chloroplast because of the lack of chlorophyll synthesis and carotenoid synthesis. On the next slide, you have the link, the biochemical link. So it's a mutant which is impaired in the conversion of 2,3-epoxysqualene into cycloartenol. So this is shown on this GCMS analysis. So you have the wild-type profile with the main peak being cytosterol, and then next to it you have a cycloartenol synthase mutant that accumulates oxidosqualene. So that's a biochemical mutant. You have the accumulation of oxidosqualene, and still the diagram shows sterol profile that is quite similar in terms of sterols, pathway end products to the wild type. So we have apparently a development-dependent albinism of cell layers in the stem associated with down-regulation and reduced activity of cycloartenol synthase. So on the next slide, we go a little bit further into the details by quantifying this data. So in black, you have the substrate of cycloartenol synthase, 2,3-oxidosqualene, and in gray, you have the sterols, all the cyclized sterols, mostly of course, the 24-alkyl delta-5 sterols, the pathway end products. And what is striking, especially in stems, which is circled with the red form, is the total amount of substrate and products is increased in the mutant compared to the wild type. You have this gray bar in wild type, flowering shoots, black bar and gray bar. So the sum of both is superior to what is found in the wild type. So there's an accumulation of oxidosqualene, so that's mutant, of course, but sterols do not decrease, especially in these organs. And so this implies a regulatory component, so something is happening in this mutant that is like a feedback loop or something, which is stimulating the activity in that metabolic segment. And we considered that the mevalonate and then the cycloartenol to cytosterol pathway, and we checked a little bit the enzymes upstream, and of course there is an enzyme called 3-hydroxy-3-methylglutaryl coenzyme reductase, which is a known enzyme that has some limiting characteristic in the pathway. And so we measured the activity of these enzymes in microsome prepared from stems of wild type and mutant. And on the next slide, it is indicated that we were able to measure a slight increase of activity in the mutant compared to the wild type at using several repetition of these microsome preparations to measure HMGR using radioactive substrate. So it is like apparently 2,3-oxidosqualene is triggering an increase in HMGR activity, and then the metabolic pathway is like cycling in a way to produce cycloartenol and 2,3-epoxysqualene, and part of it is cyclized into sterol by this weak allele. So the molecular mechanism is yet unknown, and so we are still investigating this mutant to understand a bit further what's going on in this pathway. On the next slide, we have indeed shown that HMGR is a regulatory enzyme acting on the biosynthetic flux, biosynthetic activity for the production of sterols. And we were asking this question, which is quite still unbroken question of the regulators implied in the control of HMGR, and more generally in isoprenoid phytosterol homeostasis. And so for this, we implemented a series of forward genetic approaches, and still going on in the lab in Arabidopsis thaliana, to have a de novo approach without a priori to get insights into the organization of these metabolic networks. I have a picture that maybe you know well, is the understanding of the cholesterol pathway regulation in mammals. And so you know that cholesterol is implied in the regulation of the production of sterol regulatory element binding protein, these transcription factors that are quite well known. And so the rationale of using genetic screens in Arabidopsis is that all these components like sterol regulatory element binding proteins, SCAP, INSIG, etc. There are no orthologs in plant genomes. It does not exist, so the system is different. On the next slide, I'm explaining the what we did with this genetic approach to study isoprenoid homeostasis. So we considered a mutant, an Arabidopsis mutant that you can see here on the left. So you have the wild type that has two functional genes encoding HMGR, so 1 and 2. And then we have the other guy, HMG11, so has a knockout in HMG1. So this plant is growing with solely operating HMGR2 for the production of sterols and also all the other biosynthetic segments that are branching from C15, etc. And so to identify key regulators or regulators implied in the production of sterols and possibly other isoprenoid, we did the genetic screen. So we took this HMG11 mutant line and we did the EMS mutagenesis to generate M2 families of a population of mutagenized plant. And we carried out quite difficult screening because it's based on phenotype. So it requires quite a very framed growth conditions in greenhouses, and we were looking, in fact, for second site suppressor mutations that we call into restoration of growth in HMG11 background. And in fact, the screen was also designed according to the next slide here in order to possibly eventually identify components that could be implied in the connection of both precursor pathways, so the mevalonate pathway that is generating C5 isoprenoid building blocks from acetyl-CoA, and the non-mevalonate pathway that is generating exactly the same isoprenoid building blocks from glycerylate phosphate and pyruvate. So this biosynthetic pathway is occurring in chloroplasts for the production of carotenoids, chlorophyll side chain, etc. So we did the screening and some others also targeting other genes. We were able to select quite a number of suppressor mutants. So this is to explain a little bit the phenotype of this plant. So you have on the left side up, you have WS2, that's a wild type Arabidopsis, and just underneath you have the parental line HMG11, so which has this growth deficit, and then the next series of pictures you have individuals from segregating populations of mutant, segregating families of mutant after performing backcross generating F2 backcross 1 backcross 2 families. Okay, so you can see clearly a restoration of growth in these segregating families and individuals that have also the parental phenotype. So the most informative morphometric parameters that we measured is the seed fruit filling. So you have the other series of photo. You have wild type calyx and mutant calyx, so that's the fruits, and then the suppressor, they have an increased size of calyx that is counting as a proxy for the number of seed production, which is quantified on the right side. So we next carried out bulk segregant sequencing analysis, looking for SNPs according to EMS mutagenesis, and we were able to identify one of the line, a mutation in a gene which is 81G6995. So we have isolated one allele that carries a stop gain mutation in this gene, and the loss of function of this gene, in fact, results in the phenotype that we have been able to isolate, like a restoration of growth of HMG11. And so, of course, the obvious question was to check whether the enhanced growth in this mutant was due or partially due to an increased level of HMGR2. So the only functional HMGR gene in the background that we use, in the parental background that we use. And so there are indications, for instance, in this graph is showing gene expression measurement by qPCR. So it gives an indication that you have an increased gene expression of HMGR, but quite moderate at this stage. So on the next slide, we ask the question whether this gene, which was poorly annotated until we came to that gene, and also another group came across the same gene, as you will see. So that gene is apparently acting as a regulator of HMGR directly or not or indirectly. And so it is for us a novel key player component in plant sterol homeostasis. That gene was poorly annotated as membralin in databases. And so that gene we could not outcross that allele from our parental background because in fact the HMGR is sitting quite next to that gene on the same chromosome arm. And so to be able to answer the question, what is doing a loss of function allele in a wild type background? This is what we did. Result is shown in the next slide. Yes, so the loss of function of that gene product in a wild type background is in fact, to make the story short, generating an increased amount of sterols. So it's triggering phytosterol overproduction. So you have a wild type, which is shown on this simple graph, and two of the lines that we have isolated. And in fact, this allele, loss of function allele, carrying a tDNA. So those were obtained from the Nottingham Arabidopsis Center facility that is providing genetic resources to the community, were also characterized by a colleague of mine, Takashi Shimada at Chiba University, who showed that stop-gained alleles in this gene were in fact generated lipid droplets production. And that's making the link with the accumulation of phytosterol. So the question now is, what is this gene doing, which is a new player in the game? And this is what we are currently pursuing in the lab with two PhD students. And we have a kind of working model for that, which I think is going to appear on the next slide. Yes, so that's a simple cartoon with endoplasmic reticulum of the cell that is showing HMGR, which is located in the ER, and then you have the bunch of enzymes that is producing sterols. And when you have an increase of HMGR activity, that we have been able to show this years ago by making simple transgenics with HMGR enzymes, you end up with an accumulation of sterols that are, the excess of sterol is taken by a PSAT, a phospholipid sterol acid transferases that we published years ago. And this is leading the accumulation of sterol esters in lipid droplets, as you can see on this tag. So now we have finally, after being a little bit like frustrated that you don't see any orthologous genes of the cholesterol regulators in the plant genomes, we have now this gene 81 G69N5 that is located in the ER according to recent experiment that we performed also with Takashi Shimada. You have this protein that is sitting in the ER. And so the question is how it is interacting with HMGR to go into further analysis of the mechanism of this regulatory system. I have a last slide to mention people from the group that were heavily working with genetics, particularly Claire Villette, Sylvain Darnay, Vincent Compagnon, Aurélien Blary. Pauline Debier is taking over the job with the Arabidopsis HMGR regulator. And I would like to mention also some collaborators, especially Daisaku Ota in Osaka University for the work on sterol methyltransferases. And thank you very much for your attention. Before I answer your questions, if there are any, I would like to mention a meeting. Yeah, I'm happy to share this information in the frame of the lipid maps that we have met at several occasions in lipid meetings, nationally or internationally. So there's a GERLI meeting. GERLI is a French society. It's a lipid science network in France, and the next meeting will be organized in Strasbourg, November 2025, and you will find information quite soon on the website, which will be updated. Thank you very much.
Thank you very much, Hubert. That was an excellent talk. And so maybe I could start, Hubert. The mechanism you described at the end with the novel regulator, the AT1 gene, is that known in any other species, or is that unique to the plant regulatory mechanisms?
No, the gene is conserved. You find orthologs in plants. Mm-hmm. And we are doing a little bit of more analysis to cover the whole classes of organisms. Mm-hmm. To see whether this gene has some broad importance rather than just being functionally important for plants.
Yeah, because very important pathway. And the HMG1 and HMG2, is there much known about the different functions of those, or are they meant to be redundant with each other, or what's your thoughts on that?
Yeah, there have been some studies about this redundancy, and there are some papers describing a ratio like HMGR is much more important in terms of quantitative assessment, but I'm not very sure on that, because if you have a mutant in HMGR2, I mean the phenotype growth reduction phenotype is less visible. Mm. So there are reports indicating like 70, 80 to 20, 30% proportion of importance of both enzymes. Mm-hmm. And if you consider double knockout, then it's little. You don't.
That's fine. I was going to ask what happens if you take the two of them out. Yes. So you commented about the fact that in the AT1 gene, you were finding it in the flowering shoots. I think that's correct. How do you link the lipid droplets to the flowering shoots? What's the process of the two linked together in the function of the cholesterol?
You mean in shoots?
Was that what you said? The flowering shoots, that was where you were seeing?
No. If you check the plastids or chloroplasts, they have a huge amount of plastoglobuli, according to electron microscopy pictures. And so that's another sign of deregulation of the isoprenoid pathway, because these plastoglobuli are full of tocopherols, and these compounds are also products of the isoprenoid plastidial pathway.
Okay, no, that's good. Yeah, I can see you've got a question.
But the general idea with lipid droplets, cytosolic lipid droplets, is when you have an excess of sterol esters, then they are stored in lipid droplets. But this is also the case for other organisms.
Oliver Schmidt, membralins are components, are ERAD components in mammals. Are HMGR levels higher in the mutant?
Yeah, there's maybe. I mean, the question is about membralin. So I mean, there's some identity with membralin, and there was a report about membralin in yeast acting in the ERAD degradation pathway. Okay. So now we are checking carefully, according to organs and developmental stage in the mutants, the level of HMGR with good antibodies. So we are also generating tools to study all these proteins because, well, we need good antibodies, etc., to follow degradation of the proteins, etc. And that I mean, I perfectly understand the rationale of the question. Yes, sure.
Mm-hmm. Yeah. Thank you. That's addressed that. Thank you, Hubert, for coping with a slightly tricky seminar, but I think we did very well.
No,
so,
yeah.
Thank you very much,
Okay.
Hubert.
No,
That was
okay.
a lovely, clear exposition of those complex pathways and how you've unpicked them. And I like the idea that you can work with such a visible system to see the outcomes of what you're doing. Just one last one from me. Are any of the genes you've been studying known to vary across, naturally across genetics and crops that grow better versus those that struggle? Is that something you've looked at?
That's interesting. Yeah, we had in fact we are studying this in tobacco, for instance, with a project that we had with a Marie Curie Fellowship EU project to study the effect of an enhanced sterol pathway for crops in response to abiotic stresses and stuff. So there are a number of observations indicating that an increased sterol composition or modified sterol composition is part of response to abiotic stresses.
Oh, well, that's that. But watch this space. That'll be interesting to see that develop.
But so, I mean, modifying the amount of this type of compounds is it's not necessarily easy because I mean these compounds are essential molecules for plants. So you have to cope with the homeostasis. So. Yeah, and modifying the type of sterols, I mean, the individual sterols in the profile is also, I mean, this was also achieved for a number of species, but yes, so far, I mean, no such traits have been employed yet in breeding programs, to my knowledge.
The opportunities. Well, thank you very much, and thank you for coping with the challenges. But that was our excellent seminar, and really fascinating, and it's really interesting to see where it's all heading, and you can see lots of applications. So thank you very much. And I can see the claps are working. That's good.
Thank you very much for the invitation. It was.
You're welcome.
Bye-bye.
Bye.